Yes, and we are live. Hello and welcome to this week's Intelligent Infusions Masterclass. Welcome everybody. My name is Philipp Harnisch. I'm head of laboratory here at Biotech, and this week's webinar is especially interesting for me because I do work every day with the polymers our speaker Tim Dafforn will talk about today. Before I introduce Tim, let me tell you a little bit about how this webinar series is usually going. So after my introduction, we're going to get this wonderful talk, and during the talk, you can ask all your questions in the question box, which you should see on the bottom right corner. Just click on questions, type them all in the chat, and then after the webinar is finished, we're going to pull these questions on the screen. I'm going to read them out, and Tim will casually explain everything about the polymers and about your questions and answer them. Yes. So our webinar series, we do since quite a long time now. It has been stable since 2 or 3 years for us here at Cube, and we like this very much because young and very big scientists can talk about their science here. We have a spot for everybody, everything, which is great to the public. We are happy to showcase here. So if you're interested in giving a webinar here, you can always write us. We're going to check it out and then come back to you, and you can also give your talk here in our Cube Biotech Intelligent Infusions Masterclass. But now to Tim Dafforn. His title today will be, how do you choose a copolymer for solubilizing your membrane protein target? That's a very important question. Tim Dafforn is a big guy in copolymer science. In 2009, he basically discovered, together with Tim Knowles and Michael Overduin, how to use SMA as a solubilizing agent for membrane proteins. And from this small idea, a huge community emerged. We are more than 300 people, I think. It's called the SMALP community. We meet every quarter once, and we have a conference every year where Tim is a big part of. He has been stable since 2009. And Tim is a professor of biotechnology at the University of Birmingham, so not so far away from Germany. And of course, he continues his work with SMA, but he has also interest in engineering, biology, and clinical diagnostics development. And if he's not working in his spare time, he rebuilds and competes in classic automobiles, which I think is a really cool hobby. So now to Tim, you can switch on your camera if you wish, and you can start your presentation. I will repeat the title again. How do you choose a copolymer for solubilizing your membrane protein target? Oh, we can't hear you, Tim. You have to switch on your microphone, maybe.
We go. Right, brilliant. Can you hear me now? Fantastic. Thanks for the brilliant introduction, Philip. That was fantastic. What we're going to talk about today is one of the challenges that has occurred in our area. So can you see my slides okay? Is that clear, Philip? Yeah. See the slides, hopefully. Yeah, good, brilliant. Okay, so yeah, so as the introduction has been said, we developed styrene maleic acid copolymers as a solubilized agent in 2009. And what I'm going to go through today is a little bit of a reminder of why they might be useful things to apply both to fundamental membrane protein research, but more also the applied side of understanding how to develop new drugs and new therapeutics and also new herbicides and agrochemicals, and how SMALPs can help you with this and how copolymers as a whole can help you with this. So a little bit of an introduction, because I keep finding people who haven't seen the methodologies that we use, and so it's important to show you what we do. And then I'm going to go into some new research carried out and by one of my bright PhD students, Adam Evans, who's done some work on how to screen polymers in order to optimize your purifications for making membrane proteins. So getting into the world first, I think it's always useful to define the challenge. And I've shown it here as a challenge of molecular drug discovery, but actually this is a wider challenge for everyone who works on membrane proteins. But if you think about it as a challenge of drug discovery or herbicide discovery or agrochemical discovery, what we're always trying to do is we're trying to take a protein, a target that we've identified, and we're trying to challenge it with some form of molecule, whether it's a small molecule or an antibody, an immunomolecule, and then we're hoping to get some kind of effect, and an effect that we desire, while at the same time we're trying to prevent side effects, because we want a molecule that does exactly what we want. And that's a significant challenge if you've got a soluble protein, but it becomes a really big challenge if, as is quite often the case, the molecule that you're targeting, the protein you're targeting, is a membrane protein. And so we have to add quite a lot of complexity. So conventionally, from back all the way to the sort of 1990s, what we tended to use was detergents to make membrane proteins. And I think the area of membrane protein biology has matured somewhat. And now we're aware that actually it's not always a good thing to use a detergent. And this shows some of the reasons why. I mean, membrane proteins have been evolving for 3.9 billion years, and they've been evolving to be in one place, and that's the membrane. And so I've shown here my membrane protein in blue with the bilayer there. And actually, a lot of the time, even if you transition into bilayers as a scientist, you tend to use things like DMPC, so standard single lipid bilayers. But the situation is even more complex. Not only does the membrane protein live in a membrane, but as you can see by these different colored lipids I've shown in there, it's living in a membrane which is made up of different lipid types. And actually, as we're becoming aware, membrane proteins like to associate with certain lipid types in order to maintain native-like function. So you're adding an extra level of complexity even further, which certainly detergents can't replicate. So our complex, if you like, target is getting more complex, and that complexity needs to be preserved if we really want to develop things that, as drugs attack or as molecules attack only the native protein and affect it in a way that is reasonable. Now, we can add complexity further. Very few membrane proteins live alone. Very few targets live alone in the membrane. And so inevitably, there are other proteins that need to bind to them in complexes. Sometimes the membrane proteins form in homooligomers. Many pores are homooligomers, so dimers, trimers, tetramers, those sorts of things. But also, membrane proteins like to be with other effector proteins. An example would be ramps, which are single pass transmembranes that like to sit next to GPCRs and have a really big role in modulating their activity. So G protein coupled receptors, GPCRs, their activity is modulated by other co-proteins. So you need to add those into your complex mix as well. And again, detergents really struggle here because they often break those perhaps quite weak interactions, meaning it's very hard to preserve large-scale protein oligomers. And then finally, at the bottom there, that thing which looks like a blob, a lot of membrane proteins, particularly G protein coupled receptors, mediate their responses through a soluble protein that also has to bind. So you've ended up with starting with a very simple, here's an abstract protein we need to detect. We need to develop, use it to develop therapeutics, and therefore we'll put it into binding assays, all the way through to some complex system that has, in this count, just counting up, maybe 5 or 6 different molecules all working together to achieve that native-like state. And I say, I would conject that really detergents struggle to do this. And so there has to be a better way. And so, naturally, that's what the talk really starts out with is there a better way? Oh, yeah, and I should say also, just out of interest, something which has come up recently is actually a number of drugs and therapeutic agents actually come in through the side of the membrane. And the membrane is an implicit part of the drug entry path. Again, something which detergents struggle with. So is there a way of maintaining complexity for things like drug discovery, but also for sort of more academic studies? And we're studying membrane protein systems. We want to study as close to the cell state as possible. And of course, I wouldn't ask the question if there wasn't an answer of yes. And this is, I put this in just as a hypothesis, just to really drive this home. We were at a membrane protein meeting recently, and someone said looking at a membrane protein in detergent is like putting a polar bear in a desert and expecting it to behave normally. And so what we're looking at is actually how do we put the polar bear back in the polar environment and make sure that it behaves naturally. So there is a way. And we, as was said in the introduction, discovered something in 2009 that really helped that. And that was the use of styrene maleic acid lipid particles to preserve the native environment. And that's the game we're in, really. Now, it's not to say people weren't doing something similar before. The work of Sleegar and Popov had looked at amphipoles and MSPs. But what I'm going to show you here and just remind you of is the SMALP system allows you to preserve the native environment as it is in the cell with no need to try and recreate anything. So what are SMALPs? So they're made out of a polymer, styrene maleic acid. If you're currently watching this on your iPhone, you have a couple of grams of it in your iPhone. It's an industrial copolymer made in large quantities, kiloton quantities. And it has this amazing ability that when you add it to a bilayer, it creates beautiful 10 nanometer size discs through a self-assembly process, a process that we've looked into and we continue to look into. But in some ways, it's not the process that's interesting, it's the final assembly, this supported bilayer. And around the outside, you've got a polymer cuff that supports that bilayer. Now, the great thing about it, which was unique at the time, was if you throw it at cells, it would extract directly from the cell. So there was no need to pre-extract a protein into detergent and then reintegrate it into a disc system. And that's crucial for maintaining native-like environment. So we've always talked to the SMA as being like a cookie cutter. And essentially, the way we visualize the system is here's a membrane with some colored proteins in it. And then when we add the SMA, lo and behold, you end up with discs. And we've preserved lipid environment. We've preserved the environment around the protein as the protein was evolved to be. And we've also maintained any co-proteins, any other things that are important. So essentially, by doing this process, the membrane protein itself doesn't know it's been encapsulated. If it was a human being, it has no idea because what you've done is you've cut out the membrane itself and left the protein as is. Now, what I'm going to do is just show you, I mean, this is the process we use. And if you look at the protocols on the Cube website and any other website, there's a Nature Protocols paper out that we wrote some time ago. All we do is we take the membranes. I like simple things, and this is about as simple as you get. You take a membrane, you add the SMA, you spin out the insoluble material, and in that little inset at the top above the tube is an EM showing a membrane proteome. So this is the membrane proteome from E. coli turned into discs. Every single membrane protein is in one of those discs and all the complexes. And then we purify, in our case, using HISTRAP, because most of our proteins have got tags on. You can also use FLAG and those other things. And there's plenty of details on websites like the Cube website for that. So that's our generic process, just to show you probably one of the oldest versions we've done here, but it really shows you how things work. We made a GPCR. We've made many GPCRs. They seem to be the thing that everyone wants to make. Here's one which was made out of, it was actually the adenosine 2A receptor made from yeast expression system. And you can see nicely in that SDS page, total purity. And this was a high activity material as well. So we extracted up to 90% of that material from the cells, which is something which detergent struggles to do quite often. And we maintained its activity because it was in its native lipid environment. In this case, it was in a heterologous expressed native lipid environment, but it's still a more native environment than detergent. And what was really remarkable, and it's funny, I used to go into membrane protein labs and they were very quiet, cold places because the membrane proteins would fall apart very quickly in detergent. When you put something into a polymer nanoparticle, actually everything becomes stable. So this was a paper we published back in 2015, and you can see these are GPCR. This is a GPCR, and you can see in each case on the top left, you've got an increase in stability, thermostability over the detergent equivalent. At 37 degrees, top right, you can see that it's much more stable in terms of hours, even at 37 degrees. Although membranes are actually more stable in this case, although we have found now with others that actually it matches the membrane stability. Detergent doesn't like being freeze thawed, so storing at low temperature can be a real challenge. And you can see with this case, the GPCR fell apart in one cycle of freeze thawing, whereas the SMALP continued. And it actually went on for 4 cycles, which is pretty good. And finally, at 4 degrees, you can leave the stuff. And in fact, that slow decline you see in the SMALP and membrane curve on the bottom right is actually due to bacterial invasion slowly eating your sample. So if we put some bactericide, we probably would have had a flat line. So, it was a remarkable thing back then, and it still remains so. And we could even freeze dry it. This is freeze-dried GPCR in a disc, in a dust, and you can send it. We sent it by post and resolubilized it and got the majority of the activity back. So in some ways, it was a dream come true. So that's the good news, certainly. And it is a process that's very well documented out there. And thanks to Cube, they have managed to, make sure that you have reliable materials out there. which is really important. But I felt at the time that there was only ever going to be one polymer, and that was SMA 2-1, which is the polymer which we published initially. But other people had other ideas, and rightly so. And what was really good was we started seeing lots of different polymers appear. And this is a little bit of a polymer family tree from a few years ago. And you can see different families of polymers, different modified polymers. And in fact, I myself are involved in a project with Bert Klumperman down in Stellenbosch to develop new and more optimized polymers. And this was to address some of the problems that have been observed with these. For example, some of these polymers are not magnesium or calcium resilient. Sometimes you need certain really tough membranes. You don't get as good at extraction as you expect. And some people are interested in bigger sizes. And actually, you'll see quite a lot of these on the website for Cube and other places. And so it actually began to be a bit bamboozling for people because they didn't know which polymer to use. And it was a bit of a problem of we call it seeing the wood for the trees. We now have a lovely range of polymers, but a new person entering into the polymer world would ask the question, which one should we use? And in fact, in conversations with Cube from a while back, we said it would probably quite important to have a tester pack, which they now generate, so that people could try them. And that certainly helped out a lot, but it's got more extreme. There are more polymers and more combinations, so optimization has become a bit of an issue. And you can see this here. I mean, these are from, and I'm slightly careful because we're on Cube, but these are polymers. Polymers are available from other places, and also other solutions for solubilizing materials are from other places. But obviously, Cube have mostly the range of the small SMAs that we work with, and that's very bamboozling for anyone coming into the encapsulation world. So what we decided to do was to think of a way of solving this. And a really bright student in my lab, Adam Evans, who'd come from a background of detergents and screening detergents for structural biology, decided to develop a 96 well plate format. Now we will be publishing this soon, but he decided he wanted something quick and easy that he could use to optimize his purification protocol. And to do that, he made a screen that combined both purification resins. So you'll see across the top of that diagram there a load of his tag resins of different kinds: nickel NTA, nickel IDA, nickel cephorose, and some talon, and then different polymers from Cube's polymer selection screen, and use that to optimize. And he screened things using SDS page. So what I'm going to go through now is just some of those results, just to show you that some trends. But what I want you to take home is, as an approach, it's a good way of optimizing. And we learned some things, but actually in the end, we still always optimize, even though we probably could get 80% of the way there just by our knowledge now. But that wouldn't be true for people who haven't done this before. So we applied that screening method against a whole range of different proteins, GPCRs, ion channels, ABC transports, just to see if they behave differently, and from different expression systems as well. So HEC, SF9s, E. coli, and to see whether what the screen could tell you. I'm not going to show you all the data, don't have time, but I'm going to show you some of the take-home messages. So this is just to orient you. This is what it looks like, the screen. They call it the repol screen. So this is an SDS page of the output of the material that elutes off the resins after you've solubilized it. And this is ZipA, which is a protein we use as a workhorse. It's a single transmembrane protein with a globular head. And what you can see here is we've put in the red box is the protein that we want. And you can read across. If you look at the top of that, you've got each different polymer, sulfur, SMA, SMALP 140, the various ones that are available certainly in the Cube Biotech range, but you could apply this to any polymer. And then below that, you've got each lane is a different solubilization method, a bit different purification method, or different ion chelating resin. And on the far left, we've got DDM purified material just as a control to make sure that you can see something. And what you can see there, in this case, this protein is pretty purifiable in everything, although a bit of a gap in the SMALP 300 range. But you can see that it's relatively straightforward to do, certainly something that an undergraduate could even do. In fact, we have undergraduates doing this. So next slide, a moment. So now, what are the learnings? So this is an enzyme we're working on. It's a phospholipid modification enzyme. I can't tell you what it is. But it's an enzyme we purified, we're attempting to purify. It's been expressed in coli, and this immediately shows that doing screening is important. I mean, if you look across there, the number of conditions where it works are really minimal. Out of all the conditions, it really only works in 3. It happens to work in the SMA 200s, so the SMA 2-to-1 polymer. But what's really interesting is it doesn't work in all the resins either. So if you were to look at that now, you'd go, it's obvious we use nickel sepharose because that gives a really nice band. Nickel IDA doesn't work very well at all. And in fact, the talon resin, we'll talk about this later, doesn't give anything. So this justified in one go the screening methodology. And this was actually the protein that Adam was interested in. So he was justified immediately. I have to say, as a boss, as I started in SMA 2-to-1 or SMA 200, I would have said use SMA 200. But it was nice to know in this case that I would have been correct. So now we've got an ion channel, and this shows that things can be different. So you might say, well, you know, SMA 2-to-1, that's what we started with. It works for everything. But this is not true. In this case, SMA 2-to-1, you can see the SMALP 200 still produces some material. But if you were choosing the one with the highest yield, you'd probably go with the SMALP 140 and nickel sepharose. You can see there's a nice band there, or even the dibmer-glycerol, dibmer-12 and dibmer-10 all produce reasonable material. But what's also clear here is in all cases with this protein, the nickel sepharose is the best. So again, it shows that there are subtleties here. And you might have made it using SMALP 200 if that's all you had, but you'd be missing out on 4 to 5 times the yield. And that means a lot of work. So again, it shows, and this is an ion channel, it's actually KCSA, very simple ion channel, really showing quite different behaviors. ABC transporter, you can do that. You can see the same. So this is an ABC transporter called SAV 1866 that we've worked on for a while. And again, in this case, you would go with the SMALP 200 nickel sepharose, but you can also get materials that's maybe slightly purer from the dibmer-glycerol and nickel sepharose as well. So you can see again, the screening gives you a good idea. It gets you there quickly. This is a 2-day process. So first lesson really is screening can be really helpful and it gets you there quickly. To do these things individually takes a lot of time, and we use a relatively small amount of material to do this. You can also use a Western blot approach if you have tiny amounts of material. Obviously, that doesn't tell you about purity, but it does tell you where the maximum proteins come out. And we've had some very low yielding GPCRs we've optimized this way. But what is also interesting and a real take-home message here is if you look across this one, this is get back to our zip A again, the thing which purifies in everything. And there are 2 learnings here. The first one is if you look in all the talon resin, we don't get very much material at all. And that was a surprise because talon is used by pretty much everyone in detergent space. So we were confused about that, and I'll show you something on that. We're still not sure what's going on. We have a bit of an idea. The other one is, in this case, the SMA 300 doesn't seem to work for very much. And that's a bit of a surprise to us because SMA 300 can work for many things. So I think this is something which, you know, we need to resolve over time. But the main thing to take out there is that the talon resin doesn't work very well. So we looked into the talon resin a bit more, and actually this is really interesting. The talon resin is much more sensitive to pH. And we think this is to do with the way that the his tag is coiled up next to the polymer. And you have to move the pH to much higher levels than is expected in order to release your lovely his tag from an interaction it might be having with a polymer in order to allow it to bind. So this is some work we've done. We want to bring this out, work this out further to see if this is a generic case. But it's an interesting one for us because everyone would have used talon if they'd moved from detergent into our space, and it wouldn't work. And that would explain why some people get disappointed. So my one rule at the moment is don't use talon for the moment until we sort out how it works. So is there the final question really lesson here is there one optimal polymer, which is almost the converse to should we screen? Well, 2-to-1 works for many things. So the original SMA 200 is pretty good. And you can see here, these are a range of proteins which it works pretty well. Is it universal? Well, it'll solubilize most things, but not always the highest yield. So I think in our case, if you had to give one a go, you'd use the SMA 200. But if you want to really optimize, do the screening, because it's quite possible you'll find something which might have higher purity or less yield. Or you might find something which has higher purity and more yield, which is obviously what we want. But that gives you an idea. Without the screening, we wouldn't have known that. I'd say I'm quite happy that 2-to-1 works because that's where we started. But I think it's quite nice to know that you can look across the piece with this screening methodology. So finally, in wrap up, really, it's just a bit of an overview. Just to reiterate, if you're new to SMALPs are fantastic for maintaining that complexity. We've shown that you can SMALP things up to 100 transmembranes across with 18 subunits. So it's a flexible system that can purify even the largest complexes. It maintains activities. There are plenty of publications out on that. We have GPCRs are active. The one gap is ABC transporters, which we think are a bit more of a challenge, and we're working on that. They have increased stability, and we've applied them to lots of different assays. And as is becoming more and more clear, and Cube have been doing this to a certain extent as well, you can use them for structural studies, which is very exciting. Why screen? Well, screening is a quick route to that, to enjoying those benefits you've got above. It can enhance purity and yield and function. And so using a screening approach, which we'll be publishing soon, so you've got the protocol, will allow you to get to that state much more quickly and waste a lot less time. So finally, I'd like to thank a lot of people. We've been doing this since 2009, and there's only a subsection of the people on here who've been involved and the partners we've worked with. As you know, was said at the beginning, the only reason this stuff has grown and worked well is we have a very strong community in SMALP.net, supported by Michael Overduin and people like yourselves. But it has led to a really wide-ranging collaborations, both internationally and nationally, but also with a wide range of pharmaceutical companies and agrochemical companies, all of which have worked together to this common goal of producing better pharmaceutics, understanding membrane protein biology more fundamentally, and better agrochemicals. And that's me over. Thank you.
Yes, perfect. Thank you very much, Tim. This was very insightful, wonderful talk. Really great. I'm happy that you are so happy with SMA and with screening and with different resins and all with the challenges and also all these opportunities which come up. And I totally agree with you. It's a collaborative approach. Like we're all working towards the same goal. We want to make better drugs. We want to make better medicine. We want to change also the world a little bit in this. At least this is always my motivation. So this is really, really cool. Thank you very much for this. Let's come to the questions. My colleague Camilla is going to shoot them in there. And the first question is from Mafutsur Rahman. Is it possible to incorporate a detergent purified protein in SMA solubilized lipid? I'm referring to the common workflow of MSP saponin based on reconstitutions.
So what I would say is, from our point of view, we don't do that. We don't allow any detergents apart from DDM as a control into the lab, but that's just purely to make sure we focus on the work we do. But we have worked with other groups who are keen to do that. You know, they have an established protocol for making things in detergent that they want to transfer. And we think there's a number of people have had successes where they bound the detergent protein onto a column and then run SMALP or SMALP-like materials over the top, whether they do detergents first or they add fully functioning SMALPs. It's not a very efficient process, but it is possible. But it's just, I mean, I would say if you can, go into the SMALPs first, because you're not losing any of that information from the system.
Thank you. Let's go for the next question then. Any issue with the twin strep tag? From my knowledge, strep is the most widely used tag for membrane proteins.
No, we've used strep. It works fine. I mean, the only thing we have sometimes is things bind even too well. And we've had that before. So the only hint I ever say, which is something I think everyone should do anyway, is if you're doing an affinity purification, you don't get any protein out, make sure you run the beads. And a number of times we've run beads on gels and seen where all our protein went. We just have to increase the stringency of the wash. And that's great because you get even more pure protein. But we've managed to use virtually every purification method that is commonly used and got nice protein out of it.
Yeah, absolutely. Yeah, we have the same experience with that. Every tag works, no matter, like, if it's looking for purity, of course, take a tag which is going to get you very pure protein. But yes, every tag works. Okay, perfect. Next question, please. How do you optimize the purification method after extraction? You use magnetic beads?
So we haven't used magnetic beads. In fact, that came up recently, even in the discussion with Cube the other day. And I think, you know, that would be a really good way to do that. I mean, if you think, I'm an academic in an academic lab, most of our work is single protein work. So we don't need to automate. We do have an Opentron robot we use for some other stuff. But most of the time we're what we would call artisanal. We make one protein, we do studies on it. But I think it's certainly something, I suspect some of the pharma already doing this, is automating the process all the way through. So, for example, if you're doing a drug discovery campaign, I think these structures are very amenable to that automation process.
Yeah, absolutely. Here at Cube, we nearly exclusively work with magnetic beads. It's very easy. Of course, it's more expensive than resins, but in my opinion, if you can afford it, it's definitely worth to give it a shot.
I'm a poor academic. I can't afford it.
Oh, come on, Tim. Next question, please. Could you tell us if the cookie cutter is maintained as an enclosed cutter and the lipids inside the cutter are coming out or not? Very interesting question.
So there's some really nice data by Sandro Keller on this, and there is lipid exchange, and we've published on this as well. So once you get your disc, there's lipid exchange between the discs, and someone would say then immediately, how do you know you've got your native like lipid environment around the protein? Is it not just exchanging out? Now, what we know is the protein is actually physically binding to its local lipids. It's not binding to the full leaflet. It's only binding to the local lipids. So the lipids outside that initial layer around the protein that keeps it happy, those lipids are freely exchanging. And that can be quite helpful. So I think the world's just coming to terms with the fact that you might be able to play around with the lipids a little bit by, say, adding in discs with, if you want to put in some labeled lipids, you can add disc, whether you can add disc with the labeled lipids and then do some kind of fluorescence experiments. So I think we're very much in that period where people are looking at that and playing. But the data from Sandro says that the lipid exchange is much faster than vesicle lipid exchange, I think MSP vesicle lipid exchange. So they're quite dynamic.
Yeah, absolutely. Thank you very much. Next question, please. What volume of cell lysate was used, and what is the optimal concentration of SMALP for extraction?
I can do the second bit. The first bit you might need to contact me. So because being a professor, I'm never in the lab, so I always feel a bit of a charlatan. But certainly 2.5% is what we tend to use across the board. That is too much. That's an excess. But we use 2.5% because it's guaranteed success. We found in some situations you need to back off a bit, and in DIBMA actually sometimes you need to put a bit more in. So around 2.5 is a sweet spot, but you might need to modulate. We very rarely change from 2.5. We do 2.5 and then it's all good. In terms of the pellets, send me an email and I'll pass you on to Adam and he can tell you the details.
Okay, perfect. Thank you. We're going to forward you the email of Ronnie so that we can get this going. Okay, how do you optimize buffer and soil concentration or incubation time during purification?
So we use all those were done with one set of buffers and salt concentrations and timings. And I think what we know is, again, all of those are set up so that it happens. And I think these processes are pretty resilient. I mean, if you've got a good membrane, sometimes the solubilization happens before your eyes. It's like that. Some other membranes are a little harder. And so we tend to leave them for quite a long time to solubilize just to make sure we get full solubilization. But the salt concentration, it seems pretty resilient to salt. pH, you just need to keep the pH above the pH value for the polymer, which if you're happy above 7, which you should be because you're using physiological materials, all of those are pretty stable. If you look in the information that Cube have and also in our nature protocols, it gives you some ideas on what you should use and where your space is in terms of the operational space for the polymers.
Yeah, absolutely. I would also say this is fine tuning. Start with like a standard buffer, and then just work your way from there.
I'd agree.
Next question, please. How does large scale versus small scale correlate? Also a very interesting question.
In terms of this approach, absolutely perfectly. I mean, well, when I say absolutely perfectly, that's a big claim, pretty much. I mean, we don't see much discrepancy between the two scales. We usually start small scale for this, obviously, and then immediately, the guys in the lab are going up. We generally work in coli, so the guys move right up to 12 liters almost immediately, and then work from that. And it is a direct scale. You just 2.5% for membranes at 12 liters works as well as if you've got 10 microliters.
Yeah, absolutely. It's an expensive decision if you do it, but this is why the screening approach is great. So you can figure out exactly what works for you. You can try to dilute it down and then from there directly upscale. Perfect. Okay, do we have more questions? More questions. There's a lot of questions, Tim. That's great. Is there evidence that SMALPs are compatible for cryo-EM of GPCRs, for example?
So that's an interesting one. So I have to admit, my wife is a cryo-electron microscopist, but she works on slightly bigger molecules. She works on clathrin. And we have wondered about this, and I've talked to some of the cryo bods. And I think the big issue, so several people play with these, is actually getting the initial lock on for your, once you've got your data. So locking on to do the reconstructions. You remember GPCRs are mainly in the membrane. They don't stick out very much. So it's quite hard for the digital system, the data analytics, to lock onto it and then classify. Some people use antibodies or nanobodies on the surfaces of those to give you that information. And it's something which, although we dabble in cryo-EM, we don't really purposely go to structures. There's other people who can do that. Even Cube could, definitely Cube can do that. So I think if I was starting, I wanted to develop a cryo-EM protocol, I'd start with something bigger, something like a ABC transporter. So you've got plenty of space, plenty of orientation information to do that solving.
Yeah, absolutely. GPCRs are hard targets, but I can assure you it works. If we extend SMALP to copolymers, then we have done this in-house multiple times and definitely works. Okay,
I'm well impressed.
Are the polymers compatible with native MS?
So that's an interesting one. We've got part of a project at the moment in our Wellcome Trust grant is to work with mass spec people about how this works in native mass spec. So it's a bit watch this space. There was some early data from Dame Carol Robinson that seemed to indicate that it has potential. And we supplied her some materials. This is about 5 years ago. But I have to say I've not kept abreast of the publications to see where that's gone. But I think it should be fine. And I think the important thing may be that you might need to change charge on the polymer, or just strip the polymer off and fly it down as a lipid polymer, a lipid protein particle. But from what I've seen, some of this has worked quite nicely.
Yeah, we had a conference in Paris just last month, SMALP conference in Paris, and there were lots of examples, native MS, and they classically went the route to strip away the polymer and then screen what's rest. Yeah. Okay. Next question, please. Is there a reason why you did not choose ASTI in your screen?
It's purely out of choice. We chose the one set we had. I think it can be I should have said this can be applied to any polymers essentially as an approach. But we happen to be an SMA kind of lab or DIVMA lab, and so we bought the one pack to do that because otherwise it was going to get quite big. But I think as more things come along, that more screening. I mean, I want to lean on Cube to make sure that they make some plates for this. A recent thing I went to with Pharma, there was a lot of requests. Could you make pre-made plates? So watch this space. I'll lean on Philip and make sure that he does it.
Yeah, we hear you. We have this in our minds. Okay, perfect. Next question. Thank you. Have you done any study on polymers versus cell types, for example, red blood cells?
Oh yes, so we've got unpublished work on red blood cells. Good, really good question. Solubilizes red blood cells beautifully, and actually leads to some really interesting results. If you do a microscopy of the red blood cells, you can see the underpinning architecture, for example, like the cytoskeleton. Really lovely. Works. I mean, essentially, it works with everything that's got a phospholipid. And if you can find a life form that doesn't have a phospholipid bilayer, you're probably going to get a Nobel Prize. So our view is it works for pretty much everything. The only slight tweak is, and we know this from Barry Bruce's work over in the States, is plastids. So from plants, their lipid types are a bit more resilient. So you have to use a slightly more, more penetrating polymer, and the 140i or some of the other polymers in there, the esterified ones work a bit better. But we found it works for pretty much everything. If you keep the bacteria and mammalian and insect, it's the same. They're the same.
Cool, perfect. Do we have more questions? Wow. Are there downstream applications that do not work so well with SMA 200?
Oh, that's a good question. So what have we struggled with? I mean, if you're going to, it seems like SPRs are complex, and sometimes it works and sometimes it doesn't. And in fact, I mean, Cube and others are working on polymers that have tags on that allow you to stick them to standardized chips, and that may be the route forwards. But it's not a straightforward drop it on an SPR chip and it works. We've managed to tether things through the tag, and we've done antibodies tethers, and we've also done chemi-absorbent as well. But it's funny, sometimes it works, sometimes it doesn't. That's the most variable. For optical techniques, it's brilliant because it doesn't scatter, and I'm an optical spectroscopist. So CD, fluorescence, absorbance, all those things, absolutely amazing, works fine. Thermophoresis, which some people use, that's a bit more complex. It's a membrane protein, so anything which involves unfolding the membrane protein, they actually don't unfold like a soluble protein. So some of those methods are a little bit more, they need some optimization. But in most cases, most things seem to work. Crystallization, we only have one example of it crystallizing, I believe. And so maybe that's not a route to go to. We're still playing around with that. But I'm a cryo-EM believer. So, I think crystallography was probably so much last year.
Yeah, I totally agree with you on that. For SBR, as you said, we have new copolymers directly with a biotin on it. So you can just use streptavidin chips and put on them. We have been very successful with this. It's true for the classical method if you use antibody tag or just an NTA and the his tag. This can be problematic. We are also not so super sure. We have an idea, but we don't really know why this is the case. Maybe it's steric hindrance or there are other reasons. But yes, the biotin has dramatically increased the response units we can get, especially when using small molecules. Yeah.
Awesome.
Cool. Next question, please. That's a long one. Do you have any experience with large fold concentrations of nanodist elevates? For example, concentration for SEC injection. I can get good NS10 data of crude elevates, but upon SEC separation, I don't see nice discs anymore. So I wondered whether I could have damaged the disc by concentrating my sample by 20 to 30 fold before SEC. Thank you, Thomas.
So that's an interesting one. One SEC is a bit bizarre anyway. So you often don't get, if you do soluble proteins, you're used in SEC to getting a single nice symmetrical peak. SMALPs don't do that. You tend to get all sorts of, the peak's quite compound and you just have to search through it to find the one you want. So don't expect great things in terms of that sort of nice shape. But on the other side, we've recently managed to make SMALPs turn into a gel, for example. So I think at high concentrations, like anything like this, if you get too high, you're going to start moving away from it being a dilute solution of particles, and they start interacting. And then I think you can get some really weird things happening. So we may, and you can get around that by playing around with the solution conditions, but there's no rules at this point. So we did exactly that sort of experiment and ended up with a gel of proteins, which wasn't great. We played around with the components a little bit and it got back in solution, worked fine. So I think you need to be a bit careful when you get very high concentrations, but it's not something we've seen very often.
Yeah. Thank you. Next question, please. Are there any polymer-based nanodiscs which are stable in presence of high concentrations of magnesium? What is the highest magnesium concentration that polymer-based nanodiscs can tolerate?
So there are more and more coming through that are. Dibmer was one of the first, and that has a really high level of stability in magnesium. And I think Cube are probably generating some. There's some coming through that way, and we have some more coming through as well. It's one of the known problems. But the thing I would say is if you do make it an SMA and you've got an assay that needs magnesium, just as an enzymologist, just be careful about, it says 5 millimolar, but that's way in excess. And so quite a lot of the time you can drop back so you're below the threshold, which makes SMALPs unstable and still get good activity. But it is an area that we're working on. And we're seeing, as I said, there's products out there already that seem to work quite well.
Yeah, exactly. We have a broad range of polymers, and there are polymers you can use more magnesium than you would ever need in there. So this is not an issue, but I agree with you. Just be careful. Some polymers, they're complex with divalent cations, so they can just also eat them away from your solution. And then if you need them for activity, then it can be an issue. But there are solutions for sure. Okay. Any more questions? We run out of time. I see the clock. Okay, so don't worry. All the questions we have not, or Tim has not answered yet, we will definitely email you his professional answers to all of your questions. And yes, with this, I want to thank you again for this wonderful webinar. It was really a joy. Also amazing answering of these questions. I couldn't agree more. This is fantastic to have you here. Thank you very, very much for having us. And yes, next week, on 12th of December, we have our next webinar. So if you're also interested in antibody discovery with copolymers, we have a great case study together with proteogenics, and just register. So thank you again, Tim, and see you soon.
Thank you very much, everyone, and good luck with your discs.